Hi everyone, welcome back to Lecture 8E of Useful Genetics, where we're going to recapitulate Mendel's genetic analysis a bit, sort of walking in his footsteps, asking ourselves what might he have asked, and then we're going to go through it from our own perspective, what to ask yourself when you're dealing with a difficult to understand genetic problem. Mendel first described an experiment that he crossed plants of two true breeding strains that differed in one trait. Now, in this context, true breeding means the same as purebred did in dogs. It means that the offspring have the same phenotype as the parents, that the phenotype is consistently inherited. And from our genetics perspective, we know that that means that the parents are homozygous for all of the traits that we're interested in. So Mendel crossed a tall plant strain with a short plant strain. What might he have expected in the next generation? And here's some more terminology. He called that generation the F1, which was short for first filial, where filial means offspring or children. So he might have expected that all of the offspring plants would be medium-sized, the average of their two extreme parents. Or he might have expected that they would be of various heights if he'd been paying attention to natural populations. What he probably didn't expect is what he saw, that all of the offspring were tall. They were just as tall as the tall parent. This was a surprising result, and Mendel asked himself, what happened to whatever it was in the short plants that caused them to be short? Was it destroyed in the cross, or was it just hidden in these tall plants? So he tested this by crossing the tall F1 plants. He crossed these plants with each other and created what he called an F2 generation, the second filial generation. And in this generation, he saw that although most of the plants were still tall, some of them were short. And this confirmed his hypothesis. The tall F1 plants had a hidden factor that causes shortness. So he had new hypotheses. Pea plants contain two height factors, and the hybrid F1 plant, he called them the hybrid because they resulted from mating of two different types, and they contained a tall version and a short version of the factors. Plants with the tall factors are always tall. This is what we would interpret as saying tall is dominant to short. And their F2 progeny contained new combinations of these factors, the tall and short factors that were in the two parents. So he tested these hypotheses by counting the tall and the short plants. And he found that 3/4 of the plants were tall and 1/4 were short. And this is just what you would expect if each of the gametes produced by the parent plants had a random factor drawn from the tall factor and the short factor that were present in these parent plants. Now, he didn't stop there. He did one more test, which was very, very scientific of him, to push his hypothesis, to probe it, to say, well, what's another way of making sure that I'm thinking correctly about this? He reasoned that if his hypothesis was correct, then even in the F2 generation, produced by crossing the tall F1, that even in the F2 generation, some of these plants should have a hidden short factor. Now, you should be able to calculate what fraction of these tall F2 plants, should have a hidden short factor. And the answer is that 67%, or 2/3 of the tall plants, should have a short factor. And we can see why this is true if we draw yet another very simple mating square for the genotypes that produce these F2 plants. So the F1 plants contained They were heterozygous. They had a tall factor and a short factor, big T allele and a little t allele. And when they were crossed, this produced plants with two big T alleles. They were tall. Plants with a big T, little t allele, and plants with two little t alleles. These plants would have been short. We're not interested in them. Of these plants, it's easy to see that 2/3 of these plants were heterozygous for big T, little t, which is another way of saying they have a hidden factor causing shortness. Now, how could he test these F2 plants for the hidden short factor? Well, he could have done what he did before. He could have crossed them to each other. He could have crossed them to the pure breeding short strain. Again, if there were short alleles in the tall plants, he would have gotten short plants. He could have crossed them back to the F1 plants. If he'd crossed them to the pure breeding tall strain, all the offspring would have been tall because they would have gotten a big T allele from these plants. So that would be less informative. The most informative, easiest to interpret experiment is this one, to cross them with the pure breeding short plants. And we'll talk more about this. This is called a test cross. And we'll talk more about this, not in the next lecture, but in the one after that. So now let's sort of summarize the thinking steps you need to go through to figure out what's going on in a cross. So, and to illustrate it, we'll consider a cross that Mendel did later, where he took P varieties that differed in two traits. They differed in height, some were tall and some were short, and in pod color. Some had green pods and some had yellow pods. So, what do you have to think about? The first thing to think about is how many genes are involved in this cross. And because there's two phenotypes, there's the height phenotype and the pod color phenotype, makes sense to hypothesize that there are two genes involved. You need to decide how you're going to name these genes. And it makes sense to name them for the general trait that they control rather than for tall or green. So it's easiest to remember if you name them H and C, H for height and C for color. But you can name them by any name you like that you find helps you to keep the problem straight. How many alleles are there of each gene? Well, we have two versions of each trait. For height, we have tall and short, and for pods, we have green and yellow. So there should be two alleles of each gene. What are we going to name the alleles? Well, given that this is a Mendelian cross and all of the traits that he looked at had a dominant recessive relationship, we'll name them big H and little h, big C and little c. And yes, one allele we expect to be dominant to the other, given that it's one of Mendel's crosses. What are the parents' genotypes? The parents were pure breeding, so the parents were homozygous. What will be the gamete genotype? Each parent can produce only one type of gamete, and those are the types. What will be the offspring's genotypes? Because each parent produces only one type of gamete, the offspring are all going to have the same genotype. What will be the proportions of these? In this cross, they'll all have the same genotype. And what will be the offspring phenotypes? They'll all be tall with green pods. Now, what matters in what we've just done is not these answers. The answers will differ from cross to cross. What matters is learning to ask yourself this series of questions, because these are the questions that will guide your thinking about the problem. One thing we didn't ask ourselves was whether we needed to worry about linkage. And from one perspective, we can say, well, this is one of Mendel's crosses. We know linkage wasn't a factor in his crosses. He didn't discover linkage because his alleles were all genetically unlinked. His loci were genetically unlinked. But from a genetics perspective, we can say, Well, in this cross, linkage doesn't matter anyway because the parents are homozygous. No matter how much crossing over happens, it isn't going to change the genotypes. In general, as I said previously, don't bring linkage in unless you're told the genes are linked or you find that you can't get a satisfactory solution without bringing in linkage. So what we've done, we've recapitulated some of Mendel's genetic analyses. We looked at results of an experiment, we proposed a hypothesis, we tested this hypothesis against new data, and then we double-checked it by getting even more data and testing it against that. And then we went through a list of the questions to ask yourself when you're starting to work through a problem. And going through these questions will always help you to get a functional working perspective on the problem and to proceed in the easiest way. Coming up next, we're going to talk about sex linkage, a special kind of linkage. I hope to see you there.